Good morning, everyone. My name is Frank Sasinowski, and I'm so honored to be here with you all at the United Mitochondrial Disease Foundation annual conference. I'm sorry I can't be with you in person. I'm here in DC speaking to you from my office 2 blocks from the White House. But I'm thrilled to be part of this conference. Phil Yeski has been so kind as to invite me over the years. I know so many of the professors who are there with you and so many of the sponsors and patient organizations that are there. So I feel like I would be among family were I there with you now. For those of you who don't know me, I was at FDA when the Orphan Drug Act became law in 1983, and I was asked by the Deputy Commissioner to help figure out why it wasn't working. And it was my research that led to the 1984 and the 1985 amendments to the law that made it work. And then I was responsible for drafting the implementing regulations. So literally from the beginning, I've had a hand with the development of therapies for rare diseases, of which mitochondrial diseases are almost quintessentially and universally rare diseases. So I'm so pleased to be with you today. I left FDA in 1987, joined this law firm where I am today, and I've been helping people, patient organizations, researchers, and sponsors kind of work through developing new therapies for all rare diseases, especially and including mitochondrial diseases. So next slide, please. I talked to Phil about what might be most useful for this conversation. And I thought of 2 topics that I think over the past year I've seen grow in importance and could have significant benefit to those of you who are researching new innovations to ameliorate the suffering of our sisters and brothers who are affected with rare mitochondrial diseases. The 2, I'll just tell you, the 2 are looking at a global statistical test for looking at endpoints, and the other is to looking at confirmatory evidence. And so the first topic is global statistical tests. Next slide, please. Let me just explain the background is that in rare diseases, it's very difficult to achieve what is the gold standard of having a randomized placebo-controlled clinical trial that's going to test your hypothesis of whether or not the new experimental therapy is going to be beneficial to those with the condition. And that's the gold standard. And it's obvious why. I've listed all the reasons why. I won't walk you through them, but it does make it easier for anyone trying to interpret the results if you are based upon a randomized placebo-controlled clinical trial. And so that's why then it's easiest for the FDA, for instance, to interpret the results. So that's why, if at all possible, you want to try to have a trial design that's going to be a randomized controlled, placebo-controlled clinical trial. Next slide. But there are problems. And for rare diseases like mitochondrial diseases, the RCT, the randomized controlled trial, can be a very big barrier to having a successful trial because, you're only going to have small trial sizes. You're not going to have thousands like with cardiovascular trials. There's heterogeneity, both of the disease expression. That is, how does the disease express itself in terms of symptoms or functions from individual to individual? And then the affected individuals. There's going to be great heterogeneity in any trial population. Let's say age of people who are going to be enrolled, the time since their onset of symptoms or since onset of diagnosis. So there's a lot of variability. And so all of these issues happen to make it difficult to come up with a hypothesis that you can test in this randomized controlled trial. And what we'd like to do is try to reduce the uncertainty of the results of the trial to the extent possible. And what I'm bringing to you today is a way that I think is going to be a significant advance in trying to have us achieve a higher percentage of success in our trials that are run on experimental medicines. Next slide, please. And that is that a global statistical test, what is that? Well, it turns out that the former FDA director of the division responsible for mitochondrial diseases, Dragos Roman, asked a Harvard professor, Amo Karai, and I to help organize an FDA mitochondrial workshop that was held then in September of 2019. And at that time, the lead FDA statistician for mitochondrial diseases, Dr. Yan Wang, she presented on that. I'm going to talk about global statistics and what she presented and share some of her slides. But these kind of approaches had been talked about by the FDA before that mitochondrial disease workshop that the FDA hosted in September of 2019. And so I just list some of the background to looking at the general field here on this slide. Next slide, please. And what I want to do is walk you through 3 of the slides. This is just her cover slide from her presentation, just so that I can kind of orient you. I'm almost taking you like back to the future. You know, I don't have a fancy convertible car to take you back to the future, but this is like going back to the future because we're going back to the mitochondrial disease workshop that FDA organized in September 2019. But it's really going to the future because what Dr. Wang presented could be really pivotal to your design of future trials. Next slide, please. So what she explained is that in her area, which includes things like Pompe and Hunter and MPS 1, what she said is that often in those 3 conditions, there's going to be 2 different types of functional endpoints, looking at exercise capacity at 6-minute walk test and looking at pulmonary function, often by forced vital capacity. And she said, you know, if you do a modestly sized trial, let's say you had 48 subjects randomized equally 1-to-1 between your drug arm and your placebo arm, she said she just made up these numbers. So these don't go looking for these somewhere as though they exist. They're her hypothetical. I'm sorry, I'm right near the White House and we always have sirens going by. If you can pick that up, I apologize. And it turns out that she presented this hypothetical saying, what if you're stuck with this dilemma and you had to pick one of these 2 as your primary endpoint and then make the other one your key secondary endpoint, or you try to make them co-primary endpoints and then come up with some Hochberg to be able to divide your alpha. Well, whatever you did in her hypothetical, you would come up with a very strong numerical lean favoring treatment effect of your intervention on both of these 2 key endpoints, but neither one would have been successful. So whether you would have picked the 6-minute walk exercise capacity or forced vital capacity, you would have failed. And she said, this is the problem, is it's difficult to demonstrate statistically significant results because of the small sample size, or she says the small treatment effect. I'm hoping that it's less due to the small treatment effect, but the small sample size. So even if the effect size is pretty large, the small sample size means there's going to be a lot of variability in the responses, which is going to make it difficult to really come to a conclusion that the results weren't due to chance. You're ruling out the play of chance, that it's not due to chance more frequently than 1 in 20 times. So that statistical power of 0.05. Next slide, please. So she said, well, you know, one thing you could do is you could kind of adjust for baseline values. And that's one way to increase the precision of your estimate of your treatment effect. And so she showed that just by doing that, compared to a simple t-test, you could, increase your power by like 40%. So the next slide, please. But here's the key. The key is, she said, well, but if you do a global test for the multiple endpoints, that is, you combine the 2 and you look for the effect size in both, depending upon the analysis that you would do, a ranked sum test or a combined test statistic, depending upon that, either one of those would yield a p-value that was less than a P of 0.05. That is, you would have then one positive, adequate and well-controlled trial on your experimental therapy. And this is kind of an epiphany, because it means that as a trialist, you are not forced into the Sophie's choice of having to make an election as to which of these two key endpoints you're going to put your money down on. That is, you're going to allocate all of your alpha to. And it allows you actually to be able to combine the power that you would get from both. And so the next slide, please. The beauty of this is that you basically have an ability to look at multiple endpoints. The example that Dr. Wang gave was of 2 endpoints, but there's no reason why you'd restrict it to just 2 functional or symptom instruments that you would be looking at. To the extent that the results are not correlated, you get more power in your global statistical test. That is, if, for instance, in stroke, you were looking at the NIH stroke scale and you were looking at the Bartels index, which are highly correlated, or with depression, if you looked at, so I could give you lots of examples. But if you look at different instruments that are highly correlated, well, then looking at the 2 of them together are hardly going to make much of a difference, because when you do the analysis, you're only going to be really factoring in the unique information that comes from the other instruments. And so if they're highly correlated, you're not getting much value to the global test. So the problem that the FDA has seen when people have proposed looking at a global test, and this is different than a composite. I don't have enough time to explain all that. Composites are themselves vexing, because for a composite, you'd have to have multiple components, but you have to have them of equal weight or relatively equal weight, clinically. And none of those kind of conditions or criteria for having a valid composite are important to the use of different instruments for global statistical tests. And the concern that the FDA has always had with global tests is that, what do you do with it? You end up with a Z-score, essentially. And what does that mean? How does the FDA translate that into adequate directions for use and labeling, so that the practitioner who is going to take the approved therapy can counsel a patient and talk to them about the relative benefit? Well, I've had I'm working with this global statistical test in lots of different domains, and including with rare neurological disorders. I'll give you an example. Dr. Billy Dunn, the head of the Office of Neuroscience, in one of our meetings, said, well, look, what you're really talking about is that you're converting everything to time, because in these neurodegenerative diseases, you're talking about a progressive disease that's unilaterally progressive. And so what you're really doing is talking about delaying the progression of the disease. So it's time. And so you are converting everything to a common metric that you can speak about in adequate directions for use that the FDA can put into labeling. So I think we are kind of matured the concept of how to use the global statistical test. We've overcome some of the barriers to its acceptance within the FDA. The thing I can't give you, and Phil Yeske is going to be very disappointed with me, as well as all of your esteemed professors like Richard Haas and everybody else who's there. So everybody's going to be screaming if I were there in Arizona. And so I'm glad I'm not there, so you can't scream at me. But they'd all be saying, so even though you can do this, Frank, I mean, give me an example. Give me an example of where FDA has used this, and you can point to it as an approval, so that I can feel confident that we can actually do this in academia or in industry or in patients' organizations that sponsor research. And that's the flaw. I can't give you that. So I told you I have 2 topics. And so the second topic, confirmatory evidence, I'm going to give you examples, so at least Phil and everyone else there can feel a little more satisfied that Frank didn't completely, leave you with just theoretical, avenues of improved drug development that you can consider employing. So if you can move to the next slide, that this next topic that I want to move to is is something that the FDA has had the ability to use since 1997, but until spring 2021, it almost acted like for 24 years it didn't exist. So next slide, I'll explain that. Next slide. That in 1962, I'm a lawyer, so I have to cite law. Okay, so in 1962, there was added to the requirements of a new drug that in addition to showing that it was relatively safe, that it also had to show that it was effective. That is, there was some benefit to a patient. So that was added in 1962. And the words of that statutory change said adequate and well-controlled investigations with an S at the end. So the FDA has interpreted that to mean a minimum of at least 2. And that's what good science requires, that you're going to have replication so that you knew it wasn't due to chance, even if you're excluding the play of chance in 1 in 20 times by your statistical analysis, and you're confident of that through sensitivity analysis and other ways that you've probed it. But nevertheless, science likes replication, okay, or confirmation. So in 1997, added to that law was an alternative way. Instead of 2 adequate and well-controlled studies, there was added to the law a provision that says, you know what, you could have maybe just 1 adequate and well-controlled positive study on your intervention together with confirmatory evidence that's obtained before or after you got the 1 adequate and well-controlled study. So this confirmatory evidence, and that was the way to show or bolster your confidence in the credibility of the findings of the first positive, adequate, and well-controlled study. So it was an alternative way to accomplish that. Now, the former head of the FDA Center for Drugs, Carl Peck, was the one who was behind this. He refuses. He's still living. He's out there in Central California, but it was a milestone, and I helped keep pressure on the FDA to remind them that this is a whole new avenue, and we didn't see any expression of that at the FDA. So fast forward, next slide. So after years of harping at the FDA, I was kind of like a dog with a bone. I wouldn't let this go. I said, you know, FDA, you need to explain to the world, what does confirmatory evidence mean? And my good friends like Janet Woodcock and Bob Temple would said, Frank, but we have the May 1998 guidance and we think that fully implements it. And I said, no, it really doesn't, because the key statutory phrase is confirmatory evidence. And that phrase isn't explained in your May 98 guidance. So after me saying this for decades, the FDA came out with guidance that explains what confirmatory evidence means. And the first person to ever say this was Peter Stein, who's the head of the Office of New Drugs. I'm vice chair of an organization called the Every Life Foundation for Rare Diseases. And at our annual conference at the Willard Hotel, like about 2 blocks from where I'm sitting now in Washington, D.C., he showed a slide that said, yes, we FDA recognize this 1997 law, the FDA Modernization Act, or FDAMA, and Section 115 of the FDAMA law as creating a new standard, a new way forward to get drugs approved. And then the FDA, it turns out we didn't know this when he made this startling announcement and showed slides, and this was just fantastic. We were elated in the rare disease community about this because it meant that we're not stuck with 2 adequate and well-controlled studies, or the one way that the FDA said in its May 1998 guidance is that you can have a single adequate and well-controlled study, but only if it's highly persuasive statistically, which some people have meant had a p-value of less than 0.001, and it's probably unethical to replicate the trial. So those are very high standards. So that single study standard almost never exists. So, but this gives us a way forward. And my analysis of all the FDA rare disease approvals since 1983 shows that the FDA has been flexible. But how have they been flexible? It's because they've been often using 1 adequate and well-controlled study that was positive and then finding confirmatory evidence. So what the FDA did is defined in guidance that came out in December 2019, what does this confirmatory evidence mean? Now, these are just the illustrative examples. I don't think these are meant to be exhaustive, But what it meant is that the FDA has said that there are 4 ways in which we FDA will accept confirmatory evidence. So the next slide shows us when the FDA, I think it's on the next slide, let's see. Yes, the next slide, Dr. Stein said that look, we're going to use this confirmatory evidence as our way to bolster our credibility or confidence in the findings of your single, adequate, and well-controlled study, hitting a normal p-value of p of less than 0.05. But when it depends upon the persuasiveness of your single trial, the robustness of your confirmatory evidence, the seriousness of the disease and the unmet need, which is always true for mitochondrial disease, always serious, and there's always a huge unmet medical need. Size of the patient population, the smaller, the better for getting confirmatory evidence, because it means that it's less feasible that you're going to be able to conduct another trial. So he said, so for mitochondrial diseases, what he said is, we will look at confirmatory evidence when these factors are met. Well, they kind of almost fit mitochondrial disease like a tailored glove or kind of a well-designed shoe. So what were the types of confirmatory evidence that the FDA has articulated in this December 2019 guidance? It says there are 4 different types. It's when you have data that's coming from a related indication. Let's say the same drug is approved. One example the FDA gave is that Rusty Katz, when he was at FDA, approved a drug for a rare pediatric movement disorder called Lennox-Gastaut. And he relied upon studies that were done in adults with partial onset seizure disorder. They weren't enough to get an approval for the adult indication, but those studies were persuasive enough to be confirmatory evidence in a related indication. Second way is mechanistic information. That's what I'm going to talk about the most today, so I'm not going to dwell on it now. Third is analyzing data from well-documented natural history to show that the findings that you had on your randomized control trial, when you're looking at it, your experimental arm versus the placebo control arm, that if you then look at well-documented natural history and you see the well-documented natural history kind of matches what your placebo arm did in your RCT, that gives you more confidence that the reason that there was a difference between your experimental arm and the placebo arm wasn't that the placebo arm acted aberrantly, atypically. No, that the natural history will tell you, oh yeah, that's exactly what we saw happen with your placebo. So therefore, the between group difference is due to the benefit of your intervention. All right, and the last thing was, what if you have adequate, well-controlled studies from other members of the same pharmacological class? And there are lots of examples like that. FDA approved irbesartan and losartan for diabetic nephropathy on the basis of one trial, each one with a P of just barely less than 0.05, but it's because both are the same class of pharmacological class of compound. The FDA said we can approve each for the other. All right, next slide. So confirmatory evidence and focusing on compelling mechanistic information. You're going to understand why when I get to my examples. And that there's compelling mechanistic information. This is the language. I'm not going to read it. I wanted to provide it to you because I know that UMDF will make full access of my slides to you, so you can read these at your own leisure later on. But you can see what the FDA was saying is that you can have mechanistic information that can come from either pharmacodynamic measure, biomarkers in your clinical trials, or you can come up with information that comes from animal sources. So, or a combination of the two. So next slide. So here we were very elated. I was personally elated that the FDA has finally recognized that the 1997 law exists, and the FDA recognizes it exists, and as a regulatory body is going to embrace it. And so when this came out in December 2019, I wanted to party. But nothing seemingly happened until about spring of 2021. And then all of a sudden I started seeing across every review division, and I wouldn't be a bit surprised if there was some internal training that went on sometime in late winter 2021 or early spring 2021, because all of a sudden across all review divisions, I started seeing evidence that FDA was saying to people, even in pre-IND meetings I was going in on, and FDA would say, if you're a rare disease, you can probably only do one trial. If you do one trial and you get a positive p-value, you're going to need confirmatory evidence. You should start planning now for what that confirmatory evidence is going to be and start talking to us about it. So we started seeing this across the board. And I wanted to really focus, I include in the slide materials detailed examples from two different review divisions. One is the division that is responsible for looking at liver diseases. And so I included two examples of those from the approvals in July and September of 2021. You can see, and it turns out that they're very similar. And I have all those materials in these slides, and I'm not going to walk through them because I want to focus on what's more important to you. And that is the Division of Rare Diseases and Medical Genetics, which is responsible for mitochondrial diseases, that they had two examples, Zokinvy and Nulibry. And so I wanted to show you those examples. And just so I wanted you to know that the Center for Biologics, if some of you are going to be looking at, let's say, gene therapy, which is in the OTAT, or a cell therapy, stem cell therapy, and that's also in OTAT over in the Center for Biologics, that the Center for Biologics has examples. I included only the rhythmic example in my slide deck, not the stratigraph, but they're very similar. And so you get the idea that it applies to OTAT as well. I was really focusing more for us today on the Division of Rare Diseases. So we're going to spring ahead. So if you can show the next slide, but we're going to pass over this one, which is one of those in liver disease, and also Liv Marley, which I had the pleasure of helping on, which is also another liver disease. And then the takeaways, you can see that those. Now, I wanted to get more to these examples. Now, this is progeria, terrible disease. And if you want to think of it, if you don't know of it, it's kind of like accelerated aging. And with progeria, what the sponsor did is it had not a randomized controlled trial, by the way. So it's such a rare disease and so devastating that the sponsor was able to do a study in looking at survival against matched historical controls. So they only had one study. You know, they might have called them, actually given them two different study numbers, but I mean, it was really one study because they called it the Prolon 1 and the Prolon 2, but it was really a combined analysis of these pooled patients. So it was really like one study against natural history. So how do you then get to confirmatory evidence? And what they did is they looked at the mechanism of action, the accumulation of progerin in the internuclear membrane. And so that's the pathophysiological genesis of this disease. Now, usually when you're looking, when I start saying words like that, somebody out there is going to say, oh, I know what Frank's talking about. He's talking about accelerated approval. No, no, no. After the days of Aduhelm, I don't know, even though I helped create the accelerated approval pathway for the AIDS crisis in the mid-'80s when I was at FDA, basically the brainchild of my good friend Bob Temple, but I had a small hand as a young pup back then. And it turns out, I'm not talking about accelerated approval, because after Aduhelm, Alzheimer's accelerated approval, there's a lot of criticism of the accelerated approval pathway. So we'd rather not go through accelerated approval. Let's just go for a full approval. But if you have a biomarker like progerin, you know, and you can look at that in your clinical trials, that can be the mechanistic information, that can be the confirmatory evidence that can support your positive finding on your functional or symptomatic benefit. Here it was the ultimate symptomatic benefit. It was survival. Next slide. And I want to show you how similar this other example of Nulibry was very similar to that, in that, again, this was another case in which you were talking about mortality advantage. So again, a very significant finding, but it's very rare. And so here again, the FDA, these are the words of the FDA approval documents. You know, this is talking about how this is looking at the pathophysiology of the disease, and it also was looking at some animal models, and because you can look both at pharmacodynamic biomarkers and animal models, or both. So you should be talking to the FDA very early on when you're designing your studies. instead of talking just about, what's the right dose, what's the right population, how many subjects, what's your randomization, what's your primary endpoint, what are your key secondary endpoints, how you're going to allocate alpha, how you're going to handle missing data, all those traditional things you talk about, it would be good if you could also start talking about what's the confirmatory evidence. If this trial ends up being positive with a p-value of less than 0.05, what's going to be the other thing, the confirmatory evidence from that 1997 law that the FDA finally recognized in December 2019 that it exists. But what is the quantum of evidence that you're going to have? Is it going to be pharmacodynamic biomarkers from your clinical trials? Is it going to be animal models? Is it going to be biomarkers in your animal models? So think about that from the very beginning of your program or wherever you are in the program. Next slide, please. So again, there were some common takeaways. I've kind of walked through some of them. There are some that are more fun than others to talk about if we had more time, like the fact that with Nulibry, the FDA was convinced that you could look at a peripheral biomarker that was tracking a CNS effect. I mean, that's really tough to do. When you can look at a peripheral you guys all know Fabry disease. I mean, one of the big problems is that you have to really look at the tissue organ, like the substrate GB3 build up in the renal capillary endothelial cells. So, it's not good enough to just look at GB3 substrate levels in the urine or blood, or look even at lyso levels in the blood. That the FDA really thinks that what's important is looking in the tissue of the organ that's of interest, that's impacted. Well, so I think this was really, so there are lots of additional things to explore in these examples, but I just wanted to just touch on this as a concept because it should inform your thinking about how you're going forward with your study designs. Next slide, please. And this was the example from the Center for Biologics from OTAT, its approval. I'm not going to go into this one. Next slide, please. So what I wanted to just conclude that these approvals are kind of evidence. I can't give you approvals for the global statistical test. I apologize. Sorry, Phil. But I am giving you examples from global statistical tests. So I think I'm giving you something that's really hard, good regulatory science that you can kind of really sink your teeth into because there's meat on the bones here. And so we can talk more about this at some other time. But I just want to say that one of my colleagues, Charles Raver, who we stole from the University of Maryland Neuroscience Lab, where he had been for like 10 years, and he got his law degree, so we brought him into our law firm. And he spoke at a conference on Niemann-Pick disease that was in Arizona, ironically, in May last month. And he asked, what do you think about using biomarkers as confirmatory evidence? And the senior FDA official who was representing the agency at that conference was enthusiastically supportive of the use of biomarkers as confirmatory evidence. So I'm just giving you more evidence that, not even citing to the examples, I'm showing how current. I mean, this is where things are moving. And I think that it would be a mistake almost for you not to consider how you can try to integrate this kind of approach into your programs. So I've given you the two examples of things that I think are really important to help accelerate your drug development programs. One, is the global statistical test, and the second is looking at confirmatory evidence. Next slide, please. What I wanted to leave you with is that last May, so May 2021, Pope Francis gave me a Heroes Award for the work that I've done on rare diseases. And I asked the Pope, I explained to him about things like Chagas disease, which kills more in his native Argentina than does malaria. And I had helped get the first drug approved for Chagas disease. So he was very interested in, and the Pope, I asked him then to bestow a papal blessing upon all of our sisters and brothers who are affected with rare diseases and all of us who work to alleviate the suffering of those affected. And he graciously, gave me that blessing. And through that, the way things work at the Vatican, it's very strange, is that when a person is there, for instance, if you held a rosary in my hand, it would be blessed. And you can carry that and then give it to someone else as blessed. Well, in the same way, there's this communicative property of blessings, papal blessings. So he did give that blessing to everyone who is affected with rare diseases, including those with mitochondrial disease, and to all of us who work to help those with these rare diseases. So I just wanted to let you know that you all have a papal blessing. And with that, I'll close. Thank you.